So, great pleasure to welcome Professor Carl Goodyear, who's going to talk to us about epigenomic profiling rheumatic diseases. Carl is Professor of Translational Immunology at Glasgow University and works at the interface between basic and translational medicine in the areas of precision medicine. So thank you very much, Carl.
Thank you. So good morning, everyone. I'm absolutely inspired now to do some mitochondrial research, and I actually do think it'd be important in rheumatic diseases, so thank you. So I'm going to shift gears, and I'm going to focus on one disease and get into the ideas of epigenomic profiling in rheumatic diseases. And I thought I'd sort of give you a bit of background in the rheumatic diseases to talk about where we're coming from in this regard. And it's partly to do with the fact that over many, many years, and with all the experimental medicine studies going on, everyone thinks about rheumatic disease and rheumatoid arthritis as one sort of particular disease itself. But actually, when you look at it, it's very heterogeneous, and every individual reacts in a very different way, responds to drugs in a different way, and I'll talk to you about that. So what we're really seeing is a picture. It's, yes, people think about it being in the joints, but it's not restricted to joints. This is a very systemic disease that has multiple different ways of presenting. And we try and treat it as one particular disease, but I think we need to be smarter than that, and that's where I'm getting at and what I'll talk to you about. And it really is trying to undefine the molecular endotypes that we're seeing right across the spread of individuals, and then trying to understand that and see whether we can harness that to figure out how we treat people when they come in. So just general terms, there is no permanent cure for rheumatoid arthritis. Essentially, all the drugs we have, and we actually have quite a lot of drugs that work really well, they just manage the condition for the most part. Some individuals will go into remission. I'm going to talk to you about that later on. But most of it is about controlling inflammation. This is an autoimmune inflammatory condition that really drives itself, self-perpetuating. And yes, we can sort of dampen that down and get people into either low disease activity or remission, but it's still there because we're attacking self. So how do we understand that, and how do we get into all the various different complex aspects of it? I was talking to you about drugs, and what we see is that although we've got a range of drugs, and these are synthetic DMARDs, and DMARDs are disease-modifying antirheumatic drugs. We've got synthetic ones, and we've got the new biologics or the antibodies, either to a TNF, wiping out your B cells with rituximab, etc. They work, but to be honest, only around about 40% of patients really have a good response to these therapies. So there's a lot of people where there's a complete unmet clinical need here, and we need to understand why that's happening. And I'm going to talk to you about what the standard of care is in a minute, but to encapsulate that, what you have to think about, this is a disease and a pathology that is constantly ongoing and reinforcing itself. And if you treat someone early with one particular drug, it's possible that that drug itself will have an influence on the evolution of that pathology. And therefore, if you start with one that might not work, when you come to another one down the line that could have worked at that initial time point, it will no longer work because disease has evolved. So these are the things we're trying to understand and really pick apart. So this is an idea. This just illustrates the fact that there are a range of different treatment options. We've got B-cell depleting, and methotrexate is sort of this gold standard that underpins everything. And then we have other things such as anti-TNFs that come along. And these are just clinical trial data. And when we're looking at ACR, what we're looking at is a 20%, how do I phrase this? 20% better disease outcome. So you've got 20% better after getting treated, 50% better or 70% better. And you can see that no matter what combination you use, the profile's all the same. We don't understand why certain people respond in a certain way. And therefore, are these the same individuals? You can't go back and retreat them at that one particular point with a different drug to understand. So it's very complicated in terms of trying to get that aspect of how people respond. So I told you I was going to sort of illustrate this in the terms of treatment strategies and what's going on. And for the most part, a bunch of rheumatologists sit around, they look at the data, they've got a guideline, and with NICE, we've heard about this, there are very defined guidelines about how you will treat someone. And usually what happens is if you get diagnosed with RA, this is early RA, you'll come in and you'll go on methotrexate, you'll go on a conventional DMARD. And then if you're lucky, about 3 months later, you might have an appointment. If that's not working, you'll get escalation. 6 months, you might get combination therapy where you get methotrexate, sulfasalazine, hydroxychloroquine. If that's not working, 9 months later, you might come back. And what happens is by 12, 24 months, etc., you might go into a biologic. And you'll get intraarticular injection of steroids during this point. But this is the the standard process in the UK. This is what you'll go through. It's just based on the guidelines. And what we see is that, for the most part, there's various different ways of looking at response criteria, but good response are only about 44%, like I said. Yeah, okay, you can get people better by 20%, around 64%, but this is not great. These are people who have debilitating active disease, fatigue, depression, etc. It's not a nice situation, so we're not doing very well. There are ways of changing this, and a very clever colleague up in Glasgow of mine, Duncan Porter, did a trial called Ticora, and basically all he did was tighten the visit numbers. And basically what he said is, if I can do tight control, if I can get someone in at baseline, give them methotrexate, I'll just bring them back 1 month later. And if I do that, can I manage the disease a little bit better? So these just shifted things to the left in terms of treatment. And what you see is that rather than a 16% remission, if you get people early, you can get 65% remission. Now, whether that proceeds long-term is another matter, but just by controlling the disease very quickly with one particular drug, methotrexate, you can have a big impact. But we're still not, even if we're 65%, you've still got 35% of people who are not responding and still have active disease. So lots of things happening, and we're not there yet. But what we do know is that, again, because this is all based on guidelines and how you would treat, and over many, many years, and it does work, what doesn't really drive treatment decisions is any molecular or even clinical classification. We know that disease is heterogeneous, but we don't really feed that into how we would treat individuals. And we have no predictive biomarkers in terms of the natural history, response prediction, toxicity, et cetera. And that's where I'm coming at now. And so that's the background. That's what we're trying to understand. So how do we sort of start to answer these questions? And I'm going to sort of touch on 3 different examples. And you'll have seen things like this before. I mean, basically, can we take this heterogeneous population and understand, before we ever give them any therapy, who's a non-responder and who is a responder? And can that therefore inform what we do? And you're thinking back to that treatment guideline, can we actually skip sections to say, okay, we have to follow the guidelines within reason, but if we know someone's not going to respond to those early drugs, can we skip that? Maybe get them onto combination therapy, maybe get them onto biologics earlier and really have an impact. And that's the goal of what we're trying to do. So many, many years ago, we set up the Scottish Early Arthritis Inception cohort, where we had people who were diagnosed and they came to see their consultant for the first time. They'd not been given any therapy, and we enrolled them. And we got a whole range of different samples, massive clinical characterization. We followed them up 6 and 12 months with all the sampling. We followed them up further now, and we biobanked all these. And we've got just over 1,200 patients. And the question is, what can we do? That we had multiple different questions, and I'm going to talk about this whole profiling to understand clinical response. And we've looked in lots of different ways, and we've looked at whole blood transcriptomics. We've done plasma and serum for proteomics, metabolomics, et cetera. We've done flow cytometry, and we've done PBMCs for epigenomics, and that's what I'll talk to you about soon. So that's transitioning now into this epigenomic landscape and signatures in early RA. So there's loads of different ways you can imagine to look at epigenetics. And people talk a lot about DNA methylation, histone modifications, non-coding RNAs. What I'm going to talk to you today about is chromosomal conformation loops. This is the architecture of your DNA. So rather than it being, if we think about DNA methylation, a lot of people look at the extent, the level, how much do we have? For the most part, you can think about these chromosomal loops as being present or absent. Are they there? Are they not there? And that's the type of assay we're looking at. And what these are, basically, is when we're looking at, and like I said, it is your architecture of your DNA itself. Most people tend to think about your DNA as this nice linear piece, because that's how we think about genes. But it's all folded up within your cell, and that folding is massively regulated. And therefore, what you will have is these chromosomal loops, which, proteins drive it, sequences drive this, and they'll come in, and you might have a scenario, this is very simplistic, but you can get the idea that you've got a gene here which is active and has normal expression because this is the loop that's formed. But what might happen is you might get a couple of different loops, and this might bring in a super enhancer or something else, or repressor, and therefore you get abnormal expression. It's about understanding this that might give you an idea about what's happening at a cellular level based on expression profiles. But can you harness this to really understand profiles in disease? And you can ask all the various different questions you have. So we can do this. So I work with a company called Epigenomic, who have an algorithm that can predict where these loops would potentially form within the genome. And what you can do is you can take your loci of interest, or loci, depending on how extensive you want to be, and you can look at that and you can predict where loops might form, so you get various different sequences, and we know where these loops will form. And if you know that, you can design arrays, you can design probes, etc., to go and characterize this. So, getting into some of the stuff we've done, the question we had was, can you predict whether someone will or will not respond to methotrexate? And we used, again, the cohort I introduced you to, which was the sera cohort. And we're looking at PBMCs, so this is total PBMCs. This is all your immune cells. So we're not defining a population, we're just looking globally to say, okay, can we characterize that epigenomic profile at this particular level? So this was done many years ago now, and technology's moved on. But at the time, what we had is when we were looking at this, we couldn't do all the various different chromosomal loop combinations throughout the entire genome. What we had to, based on the technology and the platform we had, was we ended up choosing 123 genetic loci. And this was based on what we knew was associated with RA previously. So we used an informed decision pathway to say, okay, which genes should we look at? And these are all the genes we looked at. The larger the genes are, in terms of this little word cloud, the more loops they had. It's as straightforward as that. And what happened is we did in silico prediction of all of these. We got our chromosomal conformational loops, and we had just over 13,000 probes. And you can see that it's somewhere between 99 plus or minus 64 loops per loci. So there's lots going on. I'm going to run through this based on time, and because I don't want to get into all the bioinformatics. But basically what we had is we have a discovery cohort where we had non-responders, responders, and healthy controls. And we want the healthy controls to get rid of the non-RA signatures. And from that cohort, we went back to their baseline samples. This is pre-drug, but we did have the 6-month and the 12-month follow-up. So we knew who was a responder and non-responder at clinical level. And we said, okay, if we do that and run these arrays, what can we do? And these were just the characterizations. So we had people who were very good responders and non-responders based on our clinical measures. Long story short, and this is all published, is that by doing this and looking at this, when we did the array, which had those 13,000, we got 922, which segregated according to the numbers you can see there. And then we did various different bioinformatics, and we came up with 30 potential loops that could discriminate responders and non-responders. Then create a validation cohort within the same sera population. And what you can see here is this is just the clinical response. This is CDI, clinical disease activity. And you can see this is where they start, this is where they end. Responders are responders. And again, what we see is when we do this, we basically ended up with 5 loops that really could discriminate. I'll show you the data on the next slide in terms of how that just works out. But what we saw is we found 2 loops at 2 particular genetic loci. And obviously, they happen to be immune ones, because that's what I chose. I'm an immunologist. But these ones corresponded to non-responders, and then we had these 3 which corresponded to the actual responders. And basically, when we did this, and this was the validation, we did a blinded validation cohort, what it boiled down to was that we could get an area in the curve of about 0.9. So we could predict whether someone would or would not respond to methotrexate with 90% certainty. That's what it boils down to. So it works really well. This is just a nice schematic to sort of illustrate where these loops were forming. And it's not always necessarily within the coding region. It can be outside in the regions where, again, you could think about some of the regulatory elements, enhancers, super enhancers, et cetera. So there's a lot of biology going on here that we can actually start to tease apart. But this was the signature, and I think that's the important thing I want to stress here, is that we were never going for one particular marker, one gene, one SNP, et cetera. What we were looking for is a signature that corresponded to whether you could or could not predict whether someone would or would not respond. Having said that, because I do a lot of other stuff in terms of immunology and trying to understand fundamental biology, I was interested in, okay, well, we've identified these, but what can this tell us about what's going on at a disease level, immunopathology, et cetera. And there was a nice paper published around about the same time we were doing a lot of this work, where they were looking at EQTLs and trying to understand what was going on in RA. Long story short is that we were able to map, these got the EQTL data that was publicly available, we looked at that, we had our chromosomal conformational loops that we defined and we knew the regions. And what we could do was map where those chromosomal conformational loops compared to the EQTLs. And what we saw is that in responders, our chromosomal conformational loops mapped beautifully to areas where we see EQTLs. And then in the non-responders, there weren't any EQTLs at all in those regions. And that's now starting to allow us to tease apart what might be driving differences in these individuals in terms of the types of processes that are driving that particular underlying pathology. Just to give you an idea, and this is a little bit of a segue, because I've talked about these chromosomal conformational loops, and I've said that they will be important, but do we know that they really are biologically important? Hopefully you'll agree that they will be. Very briefly, I do a lot of work, especially within RA, in terms of bone disease. This is an osteoclast. Osteoclasts erode your bone. If you've got osteolytic lesions in the joint, that's what's driving it. So we looked in one particular region with, you can take monocytes from blood, you can make them into osteoclasts. What we found is that if we expose these to immune complexes, there's lots of immune complexes in RA. So this is immunoglobulin binding to antigen, creating immune complexes. What we found is that when you did that, there was no rank expression, and you need rank on the surface of these monocytes to engage rank ligand, which will make an osteoclast. That's the biology behind it. There's no transcript at all, and the protein is massively downregulated on the surface. The reason I'm showing you this is because when we looked into, and this again is based on all of the work we've been doing, when we looked into the loops, what we found is that when they were exposed to these complexes, you actually lost an entire loop. So the white is a normal scenario, the red is when you've got the immune complexes there. There is no loop there. Long story short is that a normal physiological scenario, you have this conformation around this loci, which brings a super enhancer in. You treat with complexes, and the super enhancer is no longer there. So these loops are essential at a cellular biological level, and therefore the stuff we're sort of working out within our cohorts probably does have functional relevance. So that was the early RA, and that was being able to sort of harness epigenomics to really see if you can actually inform clinical decisions. And there's a lot of work going on right now to see how we actually translate that into a clinical setting, and that's all going in the background. What I thought now is I've got two other examples, and they're much smaller. I thought I'd give you an idea about how we're trying to define the epigenome in early RA to, again, get to the question about what does this mean in terms of the pathology that's driving it, and how could we use that clinically. And then the second one is a little bit different. So what we're able to do with our cohort is we can understand, again, coming back to this point, how do people respond to drugs? And we have various different profiles that we can define. We can define responders, non-responders. These are complicated graphs, and try to track individual patients right across the cohort. On the next slide, it's a little bit easier. But we do know that there are people that do respond to drugs. So they get the drug, they respond. That's all well and good. People that do not respond, and people actually that are initial responders at 6 months, and then when you look at them 12 months later, even though they're on the same drug, their disease activity has gone up. So there are different types of profiles of patients here, and what does that mean at an epigenomic level? And this is just an easier way of looking at this, is just splitting it apart to show you the sort of the extremes of these types of responses. So these are people who are responders where at baseline, very high disease activity. At 6 months and at 12 months, they just don't have disease activity at all. And then you can have non-responders, high disease activity, and it just stays there regardless of what you treat them with. And again, these were these initial responders. Technology moved on at this point when we were looking at this particular aspect. We now have the potential to look at the entire genome, or the predicted chromosomal conformational loops across the entire genome. There's this new regulatory 3D genome array, which really does look at everything. But we did the same process, essentially taking PBMCs, you can make these 3C libraries, and then you can probe that. What we found is that when we take responders, intermittent responders, and non-responders, we can actually find signatures that will discriminate those particular groups. But one of the things I wanted to share with you is that there's actually an underpinning signature of around about 183 loops that define RA. And we've subtracted other diseases from this. So you can actually find disease-specific signatures. And then in addition to that, you can define how people will respond. And by doing that, you can really understand some of the pathways that might be driving the disease itself. And when we do this, we can look into various different aspects of the immunopathology and try and understand what corresponds to non-response. And here's one example where you can see here in this particular instance, TLR4 is sort of very associated with a lot of the markers that we look at. And we can look at various different, we tend to try and overlay as much data as we can find in terms of the genomic data and epigenetic data as well to really try and enhance what we're seeing. So you do get very neat signatures. Very short. Thought I'd just share that with you because I do think it illustrates the fact that there are multiple different ways of looking at this and using this data to really understand the disease course. Right. In the last 10 minutes, I'm going to talk to you about a different point in the disease course, and this is more in sort of people who have had RA for a while, and what this might mean and how you can help change their particular scenario. So there are a lot of people, I showed you that when you can give them drug, they will go into remission, and that's great, and you want them there, but the drug's never removed, and these people usually on, let's say, methotrexate, which is slightly toxic, it's not very nice to take. Do they still need to take that drug? If they're in remission, have we put them into a state where if you withdraw drug, they'll never respond again, or they won't relapse as such? And what we know from prior studies that was done by a colleague in Newcastle is that if you take these patients who are in drug-induced remission now, the disease activity is controlled, they're looking good, they're feeling good, but they're still taking a drug. If you take them off that drug, 50% of patients will flare, and 50% won't. And we don't know why. So the question is, could we a priori find out who would or would not flare if we remove drug? So this is this third story I was going to talk to you about. And it's this idea of can we predict flare prior to treatment withdrawal? And we set up, again with colleagues in Newcastle and Birmingham, a clinical trial, a study called BioFlare, where we basically asked this question. Not go into all the details, but basically we knew people were in remission, recruited them, and then we follow them very closely. The last thing you want to do is withdraw drug and then just come back a few months later to see if they have not flared. So the way we set this up is close monitoring to make sure if people do flare, they can get back on their drug very quickly, because we know that will actually dampen down disease very, very quickly. So as you might imagine, I'm going to talk about the epigenome again. I really wanted to find whether there were stable signatures within these individuals that would predict whether they would or would not respond. So we used the whole genome arrays that I showed you. We actually did 2 time points now, because this is the stability of signature. When do you need to take it? And we used the various different epigenomic platform, and then we had a validation cohort as well. Just to run through the data briefly, we've got flarers and non-flarers again. We had these samples biobanked. We knew what the outcome was. We went back to it. We looked at extremes, flares, non-flare. And what we found is that this is 2 time points now. And we were looking at flare remission baseline and 2 weeks after withdrawal. And we found 625 chromosomal loops that were stable at both time points that could separate flare and non-flare. And we wanted to understand whether that could be created into a signature that would give us confidence that we could actually use this in a clinical setting. So we translated this into a much larger group, and we went from that 625 down to 10 loops. And you can see here the positive and negative predictive values. The negative was great. Positive is actually not too bad as well, 0.83. So we really could get it down to a 10-loop signature. We did a validation cohort of this, and basically ended up with around about 0.84 and 0.85. So again, 85% likelihood that we could predict whether someone would or would not flare after drug withdrawal. We did try putting in clinical factors into this. So there's been a lot of modeling where people have tried to understand whether clinical parameters within these individuals could actually predict. And when you look at some of the best features, actually you can get up to 0.94 for the positive, but the negative predictive size is like tossing a coin. So it's not great. And we tried integrating these with the epigenomics, but what it boiled down to was that the epigenomic model alone was sufficient enough in exclusion of the clinical variables to really allow us to determine whether flare would or would not occur. So again, epigenomic profiling within the periphery, when we tend to think about rheumatoid as being disease of the joints, peripheral markers were surrogates of whether someone would or would not flare in this context. And you can see here, that was 0.84. Can't help myself, so we actually also thought about these markers in the flare and the remission and really tried to understand again what might be going on in these individuals. We've got individuals who clinically look the same. You give them a drug, they go into remission, but one scenario seems to be that they've got into a state that's not health, but it's close to health, and you remove drug and they won't flare. and the ones that do actually get disease reactivation, pathological reactivation. What is going on? So there's a lot of work now going on trying to understand the signatures we've found to look at the various different pathways that might or might not be driving particular disease. And that gets into this, what does this tell us about reactivation of disease? This is the closest we can get to really understanding when someone actually transitions from non-disease in RA to active disease. We will never capture that in the real world because people will have RA for a long time before they go to the GP, before they get to the rheumatologist. So this is as close as we can see to this reactivation. So I think there's a powerful study here to really illustrate what might or might not be going on. So with that, I'm not going to summarize any of that because I've thrown a lot at you, but basically there's epigenomics and the characterization of the peripheral immune system can be extremely powerful in this particular setting where we've got a systemic disease to understand whether you can actually stratify people into those who will respond to a drug, who will actually benefit from drug withdrawal, etc. And we're applying this in many different contexts now, not just in RA, psoriatic arthritis, etc., to really understand the extent that this can be spread across the different auto-inflammatory immune conditions. There's lots of people involved, which I'm not going to highlight, and obviously we've got various funders, and I will leave it there. So thank you.